Welcome to this episode of InnovaSight, the monthly webcast from Innovia Bio, where we discuss all things clinical and drug development. I'm your host, Imi Farmus, and today we're going to be discussing one of the perhaps more misunderstood and at times controversial tools in every drug developer's toolbox, the single-arm trial. I'm joined by our special guest today, Behzad Kharabi. Good afternoon, Behzad.
Good afternoon, Imi.
Good to have you on the episode. So before we kick off, perhaps I can set the scene a little bit. And for all of our participants who are joining us live, please do ask your questions in the chat. We will address them a little later in the segment. So it was late last year where the EMA released their position paper on single-arm trials as pivotal evidence. It was a document that they put out that put sort of in black and white how the agency think about single-arm trials. And in all honesty, it was the antithesis of what one would describe as a love letter. The agency's document really read and reads like a litany of concerns. And I think just to maybe highlight some brief takeaways, I think first and foremost, one can expect from the EMA to be treated in the same way as a Everton supporter walking into a Liverpool Football Club pub. You will be asked why you're there. The agency makes it incredibly clear that they see, and they continue to, I think within the first paragraph, they continue to see randomized controlled trials as the standard and everything else is an exception. And they make it very clear in their document that any sponsor submitting a SAT, a single-arm trial, will be asked to justify absolutely everything. I think some of the takeaways that I think are worth highlighting is that the agency are quite concerned around the contextualization of single-arm trials for pivotal studies. They make a really, I suppose, valid point around the endpoint selection, around the justification and evidence that sponsors are expected to put together to justify absolutely every assumption we make within the clinical trial design. And I think, you know, more than anything else, before I think there was this particular view that if we were able to show that randomization was unfeasible, we had a path. I think now very much the standard is that randomization is very much impossible. From the FDA's perspective, I mean, the FDA's, I think, not to put too fine a point on it, in a period of change, the FDA have got, I think, again, a really interesting approach to single-arm trials as pivotal evidence within the context of accelerated approval, where the agency, again, lists particular methodological aspects that they're pretty concerned about, but furthermore really stipulate the role of the single-arm trial as the exception and stipulates that, you know, for a single-arm trial to be considered, one must show that it's unethical or unfeasible to randomize, that sponsors expect a rather large magnitude of treatment effect. And there's been some recent, I suppose, movement from the agency where we've seen, I think just relatively recently, where the director of CBER wrote in JAMA around sort of their thoughts around, not on single arms trials specifically, but on a very close concept, which is target trial emulation with the use of RWE, and that the field had moved forward, but I think a lot of that remains to be seen. So I think to kick us off, Behzad, given all of the, I suppose, regulatory concerns and the regulatory, I can't think of
challenges.
Word for it. Challenges, yeah, exactly. When do you think single-arm trials are still a smart bet?
So smart bet, safe bet. You know, I think obviously, which is important for the audience. You know, we do not have regulatory colleagues here on the call, but I think we can derive our experience from a clinical and RWE perspective. You cited those two guidances, EMA and FDA. I would like to add to that, you know, that they look at the issue from slightly different angles. From the FDA's perspective, what is important to highlight is actually, as you know, SAT versus RCT is more so in the context of full approval versus an accelerated approval, right? If you read the 2023 guidance on accelerated approval, it contains actually a lot of details what the FDA wants to see. Of course, noting that I think the world knows how substantial changes the US, in particular the FDA, is going through over the last couple of months, and indeed has to be seen how their stance going to be in 2025 and moving forward. What do I mean with that? If you look from the FDA's perspective, I think a SAD still is a viable option if you appreciate, as you alluded to, Imi, the treatment effect they want to see, that, the time to end and endpoints, OS, those kind of things are not measurable in the single arm design, the treatment magnitude or R, for instance, as an endpoint, but they do expect a confirmatory trial. Right? They mean, if and they actually say it also, right? So you could do a confirmatory trial in an earlier line of treatment. You start last line with a single arm trial, but then the FDA expects from you that you would already consider or even already plan or even already execute a confirmatory trial a line earlier. So that's one avenue. So they're giving actually a lot of, I would say, tips.
Oh, indeed. And also, like, the guidance really is shifted to the point that if you are submitting down this pathway, the expectation is that you do have a confirmatory trial enrolling, if not fully enrolled by the time of submission.
So are there exceptions to the rule that you could I think they're also saying that, if there are, for instance, substantial considerations which would prevent an RCT or would make an RCT unfeasible and unethical, yes, there may be, but, this is not, where you and me could give you general guidance. I mean, this is where the companies and the programs have to look at it and engage. I don't know how many times they say, you have to engage with us. You have to engage with us early, right? The only mistake you can do is indeed you run a SAD kind of in isolation and then too late come to the FDA. So maybe there may be avenues where you could technically go for a full approval, but no one could put their hand into the fire and say that this would be sufficient. The more probable route from FDA's perspective indeed is accelerated approval with a confirmatory trial or confirmatory evidence subsequently. I think the EMA, to the point you made, typical EMA maybe takes a much more conservative, more restricted approach. Indeed, SAD maybe, but it's really not what we want to see from you guys. It's about comprehensiveness of data. It's about isolation of the treatment effect, and so on and so on. So would I today say don't run a SAD? It's hard to say. It really depends on the program. It depends on the indication. It depends on the unmet medical need. It depends on the treatment effect. It depends on what discussions you already may have had with the agencies, right? If the agencies have said to you 3 times through scientific advice, we do not believe that the single arm trial makes any sense from your product perspective, sure, you can run a SAD, but the probability that they're going to change their opinion is probably rather slim. So again, one has to look at it from a holistic point of view. So I think it may make sense if you acknowledge that if you're in the US, accelerated approval may be more the path for you, or if you're in Europe, it's conditional market authorization. And then what is the confirmatory evidence you still are bringing to the table, if that makes sense.
I am in complete agreement. I think for me, it's, and both me and you have danced the single arm tango, I think many, many times. I mean, I think a lot of times sponsors, sort of move quite quickly to starting to really conceptualize the SAT design. But really what I think is so important is prior to design, really build out the justification. It's building out the narrative and a solid and robust evidence base to really justify why a SAT is indeed the only way or an unbiased way forward with the clinical development of the asset you're intending to license or get approved.
Yeah. So to that point, I think you're raising a point, right? And that is, how have we built the evidence for the treatment effect? I mean, as you know better than I, for real-world evidence support, what was the historical control on which we have established it? One thing which is important, and it's actually for those who have read in very detail the FDA guidance, for instance, right? The treatment magnitude or the benefit of the therapy will be assessed at the time of the filing, for instance. It will not be assessed at the time when you initiated the trial. So if you took 5 years to complete your study and then you came from, let's say, very outdated assumptions and your sample size doesn't cover it, it can be that the treatment effect then may not be considered sufficient because in 5 years, maybe 2 or 3 other drugs have been approved. This can, in the European context also, for orphan drug designation and so on, can have major impacts because literally the field has moved past that point. So it's all these kind of considerations. How have you built the historical control assumptions for your study? You can technically make a pivotal study with 20 patients if you want. Doesn't mean that it's clinically meaningful, right? So have you used real-world evidence? Have you used SLRs? Have you used synthetic controls? You're a much better place to answer these kinds of questions. That the evidence is robust, I would say, right? If that makes sense.
Yeah. And I think you're touching, I think, maybe to our next point of discussion, but it's really about the common pitfalls that we've seen in our time within SAT design and interpretation. And I really appreciate you mentioning earlier I think the point, if you read any of these regulatory documents, what comes up time and time again is if you're doing anything that deviates from the norm, for the love of God, come speak to us, go engage with the regulators early. And I think one of the common pitfalls that I've seen is at times biotechs really being hesitant to do so with this sort of strange conceptualization of risk. I think other common pitfalls that we've seen are, I think, a poor use, or let's say a suboptimal use of real-world evidence for contextualization. You make the point around the isolation of treatment effect, and indeed, without context, that's incredibly difficult. As the single-arm trial is being designed, as you're considering this, the contextualization strategy is absolutely important, and really building out the RWE or the historical trial or the whatever it is you want to contextualize, whatever data you want to use to contextualize this at, that really has to go hand in hand with your single-arm trial. You can't leave it for the last minute. And I think that also goes quite nicely with the aspect of pre-specified success thresholds, where again, what will define your trial as positive or negative? You may have a historical control rate, but again, how was that derived, and ensuring that the methodology by which you've gotten to that number is absolutely sound. And I think the third part is at least for me, is thinking somehow that a single arm trial may be quicker or easier. This is still a clinical trial, still has all of the complexities associated with any other study, and they do need to be thought through properly. How about yourself, Behzad? What are some of the common pitfalls that you see?
Yeah, so I think from a clinical development perspective, it's, a single arm trial may sound a quick win, right? And it may be to an accelerated approval path, as long as you have built in your timelines and considerations, again, the confirmatory aspect of it. I think the point you raise is a very important point, which is to have the right assumptions at the start, right? And not ignore. So let's take an indication. And that there is evidence out there about certain therapies. You think that in the next couple of years, other therapies are coming to the market and going even through approval. So you can technically ignore all that evidence because you want the trial to be successful. You want that the regulators don't look at it from that. I don't think they look at it from that perspective. They look at the time you file, that what is out there, what has been approved. So how robust you establish your historical control, how robust you do, let's say, you do due diligence, and have you done sufficient evidence checking on, if I say that the overall response rate is 30%, is it really 30% and not out of sudden 50%, right? Because I just didn't check other evidences which is out there. So I think for me, the package on which you build the study, what you call the real-world evidence, if it's an SLR, if it's a synthetic control, if it's what other methodology you use, will help you later in any kind of filing, right? Because otherwise the study will be considered underpowered, the treatment effect will be considered not meaningful. Now, there are, of course, indications where it's really difficult to run RCTs, and there needs to be somewhat also some level of feasibility. If you have indications where only, let's say, 1,000 people in the world exist, and you go certain lines earlier, you may end up with such a small, it's not even rare, it's really becoming an ultra rare indication, where it's going to be very difficult to even run any clinical trial. So those may be avenues one can discuss with the agency. Generally, my experience is that operational considerations and risks are not something the regulatory agencies are very open to. It's really more the scientific merit and the clinical, unmet medical need.
Well, indeed, right. It goes back to the earlier point that, yeah, it goes back to the earlier point that, we said, right, it's the justification. It's, if the single arm trial is a house, you've got to have the robust foundations in place, which is like, why on earth are you doing this?
Yeah.
And if you don't ask yourself the deep questions beforehand, don't worry, the regulator certainly will. What are your thoughts, Behzad? Like, with EMA, they sort of put you in a damned if you do, damned if you don't scenario when they talk about external validity of the data. So a lot of trials fail before they even start due to the target patient population, the inclusion exclusion criteria. How do you conceptualize that within these guidances and within this setting?
You mean in the setting of what do I consider the control arm, or?
No, the inclusion exclusion criteria for the single arm
So
trial.
yeah, so I think what for me is always really, really important when we discuss study designs, indeed, that we have the right patient population. So, generally, let's now exclude pediatric studies and PIPs for the moment, right? And so adult, which is, very specific considerations with the PETCO, in particular here in Europe versus the US. So when we talk about adult patient populations, for me, what's really important in my experience has been that if you design a pivotal study today, right, and you say, okay, let's say I go third line, certain indication, then what you really need to be very thoughtful about is, okay, what is that third line? So I have seen, for instances, that agencies, sometimes drugs are not approved in every region of the world. So what becomes a bit tricky is that if I go in a certain line in Europe, that may not be reflective in the United States. So you have to actually discuss that, and that can become a bit tricky with the agency if they do agree that this patient population, if your assumption for the unmet medical need on which you build the sample size and the consideration for the study is actually indeed reflected in this patient population. That's one thing. And they may really come down to, after 2 prior lines, after drug X, Y, and Z, but you realize drug X, Y, and Z is in Europe not the same as it's in the US, which may get tricky if you want to run it as a multinational global study, right? Which often sponsors tend to do. Again, in hindsight, if you then run a European study on the European standard of care, right, and let's say X line, this may not be the same in the US. So you're stuck because then the FDA may come back. And we've seen this, with other companies where they said, yeah, but your data was not generated in our standard of care. For those who have followed the most recent ODAC, actually, which was, I think, a few days ago, there was, I don't remember which drug it was. And I think it was because it was partially run in China and partially run in Europe. There is another complexity now coming into consideration, which is no longer just about that you're following the same standard in the US, but actually that you haven't even enrolled sufficient US patients into the study. So it was always kind of hypothetical, from the more, do these patients follow the same standard of care? We already had negative ODACs on this matter. But now it's about, well, there were like maybe I think 24% or something of US patients in that one trial. Sorry, I forgot the name, just spotted it. But then the negative ODAC, I think it was 8-1 negative, came because they said, yeah, but there's not enough US patients in there. So again, that adds even another layer of complexity. The third one, which I think is really important, is how broad is your patient population? My experience has always been, we try to be very inclusive in studies. I know one of the most classical one is that we run HIV positivity or rather negativity in oncology clinical trials. But while I personally think it is time to move on to exclude patients, HIV positive patients, or with a history of HIV from clinical trials. The challenge then becomes that in certain indications, this may have impact on outcome, and that causes heterogeneity in the data set. So then while on the one hand, the EMA encourages that, on the other hand, they may also say, now you have a more heterogeneous data set. And you sometimes feel, what am I supposed to do? To go left, it's right, it's wrong. If I go right, it's wrong. Again, for me, the discussion with the regulatory agency on the inclusion-exclusion criteria, on the patient population, that it's homogeneous enough, that the considerations, that they actually really agree to that and say, yes, we think if you assume 30% as the overall response rate based on historical data in this patient population, we consider this acceptable. It also comes down to how you ask the questions to the regulatory agencies as part of scientific advice. You can ask very broad, you can ask very specific. They may come back and say, well, it's a review issue. But I have also seen them to be very clear and say, no, we don't think that that makes any sense. So you sometimes get with a negative answer, also very valuable insight.
I think, I can agree with you more. It's the importance of actually, taking the scientific advice meeting and really getting as much value as you can out of it. Like, when I prepare briefing books, like it's every single question almost has, an entire appendix to it with all of the evidence and things to back up the assertions I'm trying to make.
Yeah, one thing you need to be just conscious about, and you are fully aware, I think for everyone to be aware is, there's a lot of backlog right now in the agency, for instance, FDA. And I think the challenge sometimes is to get them even on a call. So what I think is more and more happening is that scientific advices may come delayed, may come in written responses.
A lot of written responses at the
Yeah.
moment.
So, and that makes it challenging, right? Because how do you want to interact with the FDA if everything is just written? Do you every single time submit a new scientific advice? It can become very tedious, and it can become very lengthy. And often in the world, even of big pharma, but also small biotech companies, waiting 3 months, 6 months, or even longer to get it really shatters clinical paths, for these small biotech companies and the environment in particular we are in. One would wish the FDA would be, let's say, more open to at least phone calls like they used to be, because I even personally remember when you are on the phone with them, you can even discuss things on the phone. And sometimes, I don't want to say negotiate, but discuss. And sometimes it's really common sense discussions you can really have with the FDA, but written responses make it really difficult. Again, these are more where you need regulatory advice, but I think it actually goes regulatory and clinical together to build the right package for, the type C meetings and type B meetings, end of phase 1, end of phase 2, and so on.
Yeah, and I think just to maybe wrap up the point as well, it's just in terms of other common pitfalls, I think sometimes with single arm trials, we, if I borrow from Maslow's pyramid a little bit, is sometimes we forget the hygiene elements, right? The absolute, essentials, i.e., having a central monitoring committee or a central assessment committee, ensuring that we're blinding assessments, really addressing all of the biases and thinking about them proactively. Which are all things I think that, we almost default think are aspects of RCTs as opposed to thinking how we apply them within SATs. So I think for the, before we get to the Q&A, maybe I think it'll be really cool to maybe speak to some practical considerations for colleagues on the call that are, are either contemplating a single arm trial or have one in play already. So I'll let you start, Behzad. So what are some of let's say for somebody that is currently considering a SAT, what would be some practical things that they should be considering right now?
So when I design SATs, I really spend quite a lot of energy, time, thought, let's put it that way, in really being very, very conscious about the inclusion-exclusion criteria, very, very cautious about the patient population I want to study with that SAT, and really put a lot of effort in, for instance, the TPP, although that's more strategic for the company, but really to do the due diligence on indication, approved drugs, also put effort in what will be approved in the next couple of years, because that will actually set the scene also for what is the expected, efficacy, which I will have to expect and compete against. Remember, things such as the orphan drug designation are product specific, right? And you have to be conscious when you have the ODD at the time of filing. So in a sense, you're trying to have a crystal ball, but which you can only have to a limited degree. But I think it's really important to double down on the efforts on what is the current accepted standard, and that drives the sample size, right? And there is easy and cheap ways to do that. I read 3 papers and think that's my ORR, and there is more meaningful and I would say more mature ways to do that. I rather opt for the latter one because I know it backfires otherwise when the study is once initiated. But it's really important in open label studies, I think we all are aware, once your study has started and the statistical assumptions are set, you cannot change them. I mean, of course you can change them, but it's going to really cause major impact on the integrity and validity of your trial. And usually it's no longer being considered pivotal, even for a SAT. So do your homework, do it properly when you design the study. Then the second thing I really do is inclusion criteria, as mentioned. And I try on the one hand to be mindful of including, the meaningful patient population, but I've learned over several organizations always being too broad causes heterogeneity, causing heterogeneity backfires. So that's not smart.
Yep.
Those are, and actually, when time allows, and I think time should always allow, I actually always, in space with statistical experts, and I'm privileged to have some in my network, really put RCT next to SAT. Study design does not exist on its own. I mean, you have to do your homework and say, is really the SAT the better approach? Or am I actually not gaining much, again, with the guidances which are out there? Because at the end of the day, I have to do a confirmatory trial anyway, right? But that's a discussion one needs to have in the bigger picture of the organization, the asset, this is not. And the third is really what is important for, if you're in particular doing late stage pivotal trial, because they're all late stage, right? It's market access considerations and reimbursement considerations. We have seen now several times, in particular, for instance, in Germany, as many of us know, that drugs have been taken off the market because the reimbursement agencies literally did not accept the single arm trial designs. So where you really then wonder, well, maybe if I look at it holistically, even though an RCT may take a bit longer, I'm gaining so much from the RCT that actually it makes not worth it to SAT. So I think for me, bottom line, do your homework, really do your homework, and try early the negotiation and the interaction with the agencies.
I think for me, right, like I'll stratify my sort of takeaways maybe into 2 different scenarios. So I think first, so for colleagues considering doing a SAT, I think, maybe I keep saying this till I'm blue in the face. I think first and foremost is really piece together, to quote Behzad, do the homework as to why a SAT is the way forward and why it makes sense and why it's the only available option. There are, it's one of many tools we have in our toolkits as drug developers. There are other designs that give you the efficiency of a SAT with randomization, actually. That's beyond the scope of this, but if I'll send a resource out to anybody interested after the call. But I think that's point number one. I think also, if you are considering a SAT, it never hurts to go seek scientific advice, be that from the FDA, the EMA, or whatever regulatory agency you're interacting with. I think if you have a SAT in the field already, and to quote Behzad, it's the train has left the station. What I suggest time and time again, and I found myself in the situation where I've come into an organization and the trials are already gone. I mean, the thing I always advocate for and always do is really to conduct a very structured and deliberate gap analysis. Really look at your trial and understand where are you, no study is perfect, be it an RCT or anything else. Where are the gaps in your evidence gap? Where are the gaps in the evidence? Where are the biases in the trial? And then what can you proactively do to address them whilst the trial is underway? And maybe to wrap up as well is with single arm trials, context is absolutely key. It's really thinking about how, not just justifying your assumptions, but how you're going to contextualize that data. And if you are going to be using real world evidence, if you are going to be using historical trials, be they from an RCT or somewhere else, to really begin doing that from the moment your trial has kicked off, or if the next best date is now. So really not leaving it until your trial's read out, really doing a regulatory grade approach to your context.
I think for the audience, it's important for us who, I think both of us, I would say we're still strong advocates for single arm trials, but we are realists to when a SAT can be feasible. And it really is the due diligence one has to do before one says, a SAT is the best option. And nowadays, I think we have agency guidances now from FDA and EMA, where both of them pretty much are saying, well, an exception. In general, no. Or if we even consider an exception only for accelerated path, and you still owe us the confirmatory evidence, right? So if you put this all together, I think for me, Imi, it's really about, or for both of us, I don't want to speak for you, but it really is do your homework, engage early on, and do your homework is not just something quick and dirty. It is something which is thorough and systematic to allow that this SAT has the best chance to survive. Would that be a good summary?
I think, as usual, Behzad, you're a man of fantastic summaries. I thought that put it together. Before we kick off the Q&A, I think actually here's an interesting question. So, given that for accelerated approval, at least with the FDA, the expectation is that you have a RCT enrolling or almost fully enrolled by the time of submission. And there's many reasons why they've taken such a hard view. I won't mention the sponsors. Do you not think, Behzad, that kind of puts, smaller biotechs at a disadvantage, not just from a financial perspective to actually get an RCT up and running, but also the operational concerns there as well?
Yeah. I mean, the simple answer is, of course. I think a yeah is maybe too trivial. I think we are in a cash-constrained environment, and actually, irrespective if it's a small biotech, if it's a large pharma. But that being said, of course, a large pharma has a different, financial window and financial opportunities to still pull off an RCT versus for a small biotech company in particular. I mean, this is not going to become a session about biotech funding. That's a whole different story on its own. But of course, it has become very difficult for small biotech companies. And also remember, what is the purpose of most small biotech companies? To pull off a pivotal trial is rather the rarity, right? And acquire substantial. And so I have a lot of kudos to companies pulling this off. But yes, it's tough if you have then to convince an agency with an RCT. I would personally wish that, but that's a personal wish, that investors would acknowledge where there is unmet medical need and would actually provide the support to biotech companies that then they are addressing with a therapy. Because, getting a therapy so far in late stage development, it has to go through so many hurdles to survive. But then if there is the possibility, and if that possibility is an RCT, we kind of are stuck from a regulatory perspective to bring the therapy to patients. That frustrates me all the time, because if you believe that the therapy A is really meaningful, that it really helps the patients, I think, why are we at the industry not in the chances?
I mean, I share your frustration as well. And one of the things that gets me is indeed, look, the SAT is one way to do this, but if a randomized trial is going to give us the best piece of evidence, there are other ways of doing it than the traditional parallel design. There's many different conceptions. So I think we've got Will on the call. So Will, do you want to pass on some questions for us? I know we're coming to time, so
no, thanks so much, guys. I think that was really insightful. We've got quite a few questions, so I'll try my best to get through them. For the ones that we don't get through, I'm sure Imi or Behzad will send a message later to answer. But the first is, how would you consider single arm designs for cell and gene therapy? Are there any specific, considerations here?
Interesting question. Yeah.
Well, for those who know, we both have quite a bit of cell and gene therapy. So cell and gene therapy products, it depends how this question is, or in which direction this question is going. So for those cell and gene therapy products, it's really from a sample. So when you're starting a single arm trial design with cell and gene therapy, it's important to acknowledge that patients are considered enrolled at the time point of apheresis, which is a unique aspect for cell and gene therapy products, specifically for autologous products, right? So that is something you need to take into consideration. I'm not sure 100% in which direction this question is going. If it's more the question of, well, is it easy to run an RCT with a cell and gene therapy product? I think it comes down to what are you comparing it to? Because at the end of the day, a cell and gene therapy product still has to meet the same standards of, showing the, sufficient level of efficacy based on the correct endpoint. So, or if it's then an RCT, it's, maybe PFS, EFS, and whatsoever, an RCT in second line DLBCL, which resulted in the approval of Yescarta. So yes, RCTs are possible, which was the ZUMA-7 study. And, an amazing study. And so RCTs are possible in cell and gene therapy. Many have done so the same, BMS and others. And very doable.
It's very setting dependent though, right? It's really like, for the trial you spoke about that, it's, they move the therapy down to the second line. It's what setting are you going to be in? So indeed, it's, if it's a single arm trial perspective, then, to quote you, you're completely spot on. Your issue there is, the time zero, the day zero. Where are you going to start the clock, and how are you going to deal with that for context?
Time point of randomization.
Yeah, exactly. And
kind of randomizing. So that's something you need to take into consideration when you're designing the RCT. So I would say you need a really good statistician.
yeah, and do the homework. But every trial is different, every setting is different, and we see a lot of changes now with how regulators, it's no mystery that the FDA is going through this big period of change, and there's been, I think, some interesting decisions over the last couple of days for a gene therapy company or a cell and gene therapy company. So seek scientific advice and really be clear on why the setting you're going into makes sense and why your design is indeed appropriate.
Yeah, okay.
Next question, Will.
Perfect. Yeah. So the next one is obviously we've talked quite a lot about the perceived risks and drawbacks, but it was actually kind of understanding why anyone would consider a SAT. In what circumstances does it bring benefits?
I mean, it depends. Well, I mean, look, there's no such thing as a bad study design, right? It doesn't exist. Every study design answers a very specific question. Typically a SAT, if you were going to do one, like the summary is like, it has to fulfill, one of these criteria, so you can't randomize. It's impossible. You expect a huge magnitude of treatment effect, or that it's unethical to randomize these issues of clinical equipoise, etc. There are other designs, but the SAT isn't good or bad. It's how you use it, and it's where you're using it.
Yeah, I fully agree with that. I think, you can also run terrible RCTs. So, the study design is just a tool. I mean, it's your homework you've done in designing the right study, I think.
And I would make the argumentation that the RCT is a terrible design in some settings. It will not work. And in some particular TTAs, RCTs are the wrong way to go, but it's all very context specific.
Yeah, I think what we are trying to advocate for, and if we're starting to repeat ourselves, is don't condemn a study design because the regulatory agencies are putting your, stones in your way. Try to work around those stones, or be at least conscious of those. That's, I think.
Indeed.
Perfect. The next one is there ever a scenario where using a SAT purely for commercial reasons can ever be justified to the regulator?
Nope.
Be justified or not just be justified?
To justify, yeah, to go to.
I think you're talking to clinical developable people and not commercial people for good reason. And for us, I don't know, when I speak for you and me, I have a lot of respect for commercial colleagues because they, in the end, do help us to bring the medicine to the patients. But for our point of view, it can only be purely clinical and regulatory considerations and not commercial.
The regulator will never accept a commercial argument for why you're doing a SAT. That's Yeah.
They may consider, but that is rather rare because they never consider operational reasons, because every biotech or every pharma comes and says, oh, we can't run this trial. It's too operationally difficult.
I mean, look, Alexion did a randomized trial in PNH. Roche did a randomized trial in PNH, and that is an ultra rare disease. So,
it is possible. Yeah,
Yeah.
it is possible. It's rare, but again, if you don't want to do an RCT, you have to have very convincing arguments, and the convincing arguments can't be because I only want to initiate 10 trials and it takes me 4 years. Then they will say, well, then initiate 50 trial sites and do it in 1.5 years.
Next question, Will.
Yeah. So if RWE is so central to SATs, why do regulators still distrust or discount them compared to RCT controls?
Again, it's all dependent on how it's used. We have experience where RWE external controls and the data that was generated from that is in labels, right? And in other times, it's really key contextualizing and supportive evidence. But it's, again, there's no such thing as regulatory grade RWE or regulatory grade data. It's all about the process you're following. As I said, the director of CBER, I always forget his name, bless him, put out a article in JAMA recently, where he's really talking about this next level stuff on causal inference. I mean, we've been doing it for years as though it's a new discovery, but target trial emulation, etc. Both me and Behzad did that way back at Kite. So as long as these things are used appropriately, whether it's RWE, whether it's historical data, it's all about presenting it in a scientifically credible way.
Perfect. Well, I think that's time for the webinar at the end. As I mentioned, there's a couple more questions, but yeah, I'll make sure we get answers to you guys over the next few days.
Excellent. Well, I thank everybody for joining us. It's always a pleasure to do these. Behzad, if you have any particular aspects on clinical development you'd like to discuss, you can reach out to Behzad at Kharabi Consulting. We'll circulate his details with the recording of the webinar. And for Inovio Bio, you can reach out to myself or any other of the Inovio Bio team. We are completely focused and obsessed with accelerating clinical development and really de-risking it through the use of very, very smart data. And for that, I think that's a wrap. So thank you so much, and thank you so much to everybody who asked questions.